Good morning. And thank you for joining today's Science, Conservation, and Humanities lecture presented by the New York Botanical Garden. My name is Charles Zimmerman, and it is my pleasure to welcome you from locations across the United States and around the world to today's webinar, A Bolete Story, 50 Years of Macrofungi. At any time during today's event, please feel welcome to submit questions by clicking the Q&A button at the bottom of your window, and our hosts will raise these for our speaker to answer at the end. Live captions can be enabled by clicking the CC button and then show subtitles or full transcript. This event is being recorded and will be shared on the NYBG lecture library. Now it is my great pleasure to welcome Dr. James Lendemer, Assistant Curator in the Institute of Systematic Botany at the New York Botanical Garden, who will introduce today's speaker, Dr. Roy Halling.
Great. Thanks, Charlie. And thanks to everyone for tuning in this morning to learn about a recent chapter in the long history of cryptogram research at the New York Botanical Garden. Fungi are incredibly diverse and beautiful organisms that are important to environments and economies. Today's speaker specializes in boletes, a group of fungi that includes porcini mushrooms. Dr. Roy Halling has dedicated his career to these fungi. His master's degree from San Francisco State University in 1976 focused on boletes of the Sierra Nevada in California, and his PhD from University of Massachusetts in 1980 focused on the agaric genus Colibia in New England. Since coming to NYBG in 1984, he has served as curator of mycology, working tirelessly to document and describe macrofungi, especially boletes, from the Botanical Garden grounds in view of his office to the mountains of South and Central America and tropical islands along the coast of Australia. He has developed and published identification resources for these fungi and mentored many students. Along with everyone else here today, I look forward to hearing more about 50 years focused on fabulous fungi. And with that, I'll hand it off to Dr. Halling.
Thank you, James. Thank you, Charlie. It's a pleasure to be here this morning. And thank you all for attending and listening. When I started looking at fungi, the place where I was in Central California at a state college, and I was interested in taking every class in botany. Well, back then, fungi were considered plants, and the professor who taught mycology was interested in aquatic fungi, not mushrooms. And so he was teaching us about aquatic fungi at the beginning of the class. And he said, we don't know anything about fungi. And so all the classmates, we all looked at each other and said, what are we doing here? And so he showed us this 16 millimeter film that he made when he was a graduate student, to show us how this marine fungus, which grew on king crab eggs in the Puget Sound, how it produced its spores. So this was before the digital age, when digital was what you did with your fingers and toes. But he was showing us how cytoplasm moved inside the fungus up into a vesicle where the cytoplasm would then begin to cleave up and form zoospores. And it sort of looked like cats fighting in a bag, but over time, the cytoplasm cleaves up and eventually the zoospores form and they break out of this vesicle. This was absolutely intriguing for me at the time in the early 1970s. And when he said, we don't know anything about fungi, at the end of my bachelor's degree, I said, well, how are we going to study fungi better? He says, I think you need to go to grad school. So what I did was I enrolled at San Francisco State University and to work with an expert on boletes. His name was Harry Tears. And I worked there for 2 years looking at boletes in the Sierra Nevada. And I had the opportunity to describe my first species, and that's that yellow pored thing on the right that eventually became known as Boletus citronipores. But there was some absolutely fantastic boletes growing in California, and a lot of them were unknown. And once I finished doing the master's degree, I said, Dr. Tears, what now? He says, well, you should go get a PhD. So I enrolled at University of Massachusetts in 1976, and specifically to work with the gentleman on the left there, Howard Bigelow. And there were two other mycologists on staff at UMass at the time, Margaret Barr and Emery Simmons. Margaret worked on ascomycetes, little tiny pyrenomycetes fungi that grow on wood. Emery Simmons worked on mold-like fungi. So as I say, it was a sublime opportunity to learn a lot about fungi. Once I got my PhD, then I had the opportunity to do a postdoc at Harvard University, working with Don Feaster at the Farlow Herbarium, which occupies this building that used to be the old divinity school at Harvard. Well, there I had a chance to look at specimens collected by a shoemaker from Brattleboro, Vermont. And he got interested in boletes because he was not feeling well, and his physician told him he needed to get outside some more. So he started working with the New York State botanist Charles Peck in Albany at the time. And in 1874, he published a paper where he reported 46 species of boletes that occur in New England. Of that 46, 22 were new species. That was the first compendium of boletes that we know about in the United States. Interestingly, what he did was he described this icon, Boletus russelii, which he named for his friend John Russell, who lived in Boston. And he attributed John Russell to be the authority for Boletus frostii, that's published by Frost in his own paper, which I thought was kind of curious way to get a species named for yourself. Let your friend be the authority. Once I finished my postdoc at Harvard, I had the chance to get a curatorship at the New York Botanical Garden. And because the NYBG had a strong emphasis for a long time of studying neotropical plants and fungi, I decided to initiate some survey programs and fieldwork in Latin America. And it wasn't a specifically targeted place, but I wanted to look at as many different geographic regions as I could. And so sometimes you end up in the Amazonian rainforest here near Huachumacari in southern Venezuela, and there are no hotels, so you end up sleeping in a hammock and trying to figure out how you're going to prepare your food and so on and so on. But more importantly, if you're studying biological organisms, especially fungi, one of the things that you have to pay attention to is how do you preserve the material that you're going to collect. If you're not plugged into electricity, you don't have a dryer or dehydrator to dry your specimens. And on the left, you can see botanists with their plant presses over some kind of heating apparatus to dry their plant specimens in a plant press. Well, you don't put mushrooms in plant presses. You have to dry them at a lower heat. And once they're dried, you got to preserve them in some kind of plastic bag or area where they won't rehydrate from the humidity and the moisture. In this case, it's an old kerosene stove that's been set up in in the jungle to dry these specimens. Fortunately, you do run across some interesting material. Now here was a small white nondescript mushroom that is a wood rotter, but what was not known at the time, that in this genus Marasmiellus, there was none known to form a vulva, or this universal veil sac at the base of the stipe. And in southern Venezuela, we found this thing, and it turned out to be an undescribed species that Rolf Singer eventually described. Elsewhere in Venezuela, I had the opportunity to go into the Caribbean range of Venezuela in the north, and I found this contorted orange cauliflower-like thing. I had no idea what it was until when I got home and I discovered it was an ascomycete, a macroascomycete. And Don Feaster at Harvard, who mentored me a few years before this, I sent it to him, and he reported that it was a species described from Southeast Asia originally. And so this particular specimen turned out to be the first report of this fungus from the New World. As I said, one of the things that I wanted to do was not to be geographically focused on any particular part of Latin America at the time. So I had the opportunity to go down near the very southern tip of South America in Tierra del Fuego. And I was accompanied by Greg Mueller from the Field Museum, and we were both interested in mushrooms that would occur in those southern Antarctic beach forests in southern South America. And while there, I became intrigued with these biogeographic icons, the Nothofagus in the broad sense. Now it's been divided into several different genera. But the thing about Nothofagus is that it harbors, or is the substrate or the host for an obligate parasite, an ascomycete called Cyteria. And that genus has been known since Darwin went around Cape Horn and Tierra del Fuego and the Beagle Canal. And this genus Cyteria, it only grows on Nothofagus. And the interesting thing about it is that wherever the southern beach occurs, southern South America, New Zealand, Australia, that parasite is there as well. So I became intrigued by how these icons, these biogeographic icons, how they were distributed and how they got around, especially in pre-tectonic situations on the globe. In the northern Andes in Colombia, I was working with Clark Overbo, and we came across this mushroom that at the time was in its own genus called Rosites, and it turned out to be totally undescribed at the time, and so we described it as Rosites colombiana, and that was in the late 1980s. Earlier in the 80s, Egon Horak in Switzerland had looked at all of the Rosites species known at the time, and where you see on that map all of those dark spots, he had known about 14 species at the time. And up in the northern hemisphere, you see number 1. That's the only species that was known in the northern hemisphere, except for one from the Himalaya in India. And so Clark and I were trying to figure out how the heck did this thing get to Colombia if everything is in the south with Nothofagus. And based on the kind of work that Horak had done at the time, he hypothesized that the origin of this genus, as it was recognized, was from Gondwana and had migrated north, crossed Wallace's line, and moved into the northern hemisphere. So based on what Clark and I could figure out, that this genus had then migrated again back south from North America across the Panamanian Isthmus into Colombia. It turns out when Clark and I were working on this, the closest morphological relative was known from New Zealand, and this Rosites meleagris was a slimy cap thing. And because these are obligate mycorrhizal mushrooms, it was very hard to understand how they could get around without moving with their appropriate tree host. As it turns out, once DNA was discovered in mushrooms, that the genus Rosites became subsumed into the genus Cortinarius, and that's where, based on ribosomal genes that were done by Ursula Peintner and others in Europe. So now all of those things that were called rosites are actually polyphyletic as a genus within the genus Cortinarius. However, once in a while, you run across things that you know from the Northern Hemisphere that are edible. And we found in Southern Colombia a chanterelle. This one is the smooth chanterelle called Cantharellus laterideus. And what we found there were just baskets full of this thing. And so Dennis Desjardins and I say, oh, let's get it prepped for cooking. And Ron Peterson and Greg Mueller described this variety as a new variety. And they said in their description of that variety in the paper that the collectors ate most of the type collection. Well, I can tell you it's one of the best holotypes I've ever tasted in my life. In the early 1990s, Greg Mueller and I had the idea that there were these oak forests in Costa Rica that hadn't been properly investigated for what kinds of mushrooms might occur there. And so we started out just looking at mushrooms in the oak forests of Costa Rica. And later on, because we worked on this project for about 10 years, we expanded it to include just all kinds of macrofungi that we could find in Costa Rica. And this was sponsored by several grants from the National Science Foundation. And when you get up into the mountains of Costa Rica, you can find these incredible oak forests. And when you walk around in there, you can find obligate mycorrhizal fungi that are associated with the oaks. And you can also find saprobic or saprophytic leaf decomposers, wood decomposers. But there was such a diversity of things that we were finding, and we had no idea what they were, that like bouquets of marasmius, for example. I mean, these are almost a foot tall. And a bunch, there's probably 100, 150 individual mushrooms in this bouquet of marasmius. So one day we were out in the bush and we said, we're in trouble. We don't know anything about the diversity of what's going on here. And so what Greg and I decided to do was to contact collaborators who were specialists in different groups of fungi, mushroom fungi. Bart Bauk from Belgium worked on Russulaceae, Rod, Tullus, Amanitas, Clark, Tricholomas, John Marati, and Cortinarius, Joaquin Cifuentes, and brown sportigarics, Ron and coral fungi, chanterelles, Timberoni, and pink sportigaric, and Agon horic and brown spored mushrooms, and Lorelei Norvell and brown spored mushrooms as well. So over a decade, we collaborated together and tried to publish the new entities that we were finding in Costa Rica. At the same time, we had the opportunity, Greg and I, to teach workshops for local students in the area, and not just within Costa Rica, but students who came from countries around Costa Rica, Nicaragua, Guatemala, Honduras, Panama. And we taught 2 workshops to try to expand the local knowledge of what macrofungi existed in this part of the world. One of the things that Greg and I found interesting was because of the tectonic history of the area, which has been dated to be in that part of the world, not very old, 9 million years, more or less, when Gondwana collided with Laurasia. But what we were seeing in some of the diversity of in these oak forests of Costa Rica were species that were already known and well described, and iconic things like frosty eye, Lactarius indigo, the Clavaria delphus, and litter decomposers like this Mycena epipterygia. So what we hypothesized was that once this landmass came above the seawater in Central America, that as climate changed, the forests of oaks moved into towards the southern hemisphere, and their fungal associates moved with them. And not just their fungal associates, but also the parasites that often occur with those mushrooms. So this leaf decomposer, Gymnopus strophilis, also develops this tumor from another fungus called Syzygospora. And it shows up as well on these fungi in Costa Rica. There was a very large amanita that occurred in the oaks forests of Costa Rica that resembled the Caesar mushroom. And the one in Europe is called Amanita caesarea, and it's an edible fungus. And this is a very large mushroom. But according to Rod Tullis, the expert on amanitas, he said, no, it's not that, but it's a Caesar mushroom. And so Rod did a little background investigation, and he discovered that there was an indigenous leader in Costa Rica in the 1500s, and his name was Garabito. So he was a local Caesar of the Huitares people in Costa Rica at the time. And so we thought it appropriate that this Caesar mushroom bear his name, and we call it Garabitoana. One of the things that Greg and I hadn't realized about the vegetation in Costa Rica, above tree line, there's this region called the subalpine in the Neotropics called the Paramo. And when you get up there, you don't find any oak trees, but there's this tree-like shrub, and we had no idea what it was at the time. Once you get inside some of that shrubbery, you can actually walk around a little bit. But when you're out there walking around, we found this bolete that we had no idea what it was associated with, because it's an obligate mycorrhizal associate. And we're trying to figure out what the heck does it grow with. And it's so abundant that you could fill the back of your pickup truck with this mushroom. And it's so incredibly abundant. So on one trip, Todd Osmundson was with me, and we decided to try a little project where we would dig up the earth ball, an earth clod underneath the mushroom, and then wash the dirt away, isolate the roots, and you can see the roots, root tips up there in the upper right. And if you make a section of those roots in the center, bottom center and the lower right, you can see that those root cells are filled with fungal hyphae. And it turned out that in consultation with a plant specialist at the New York Botanical Garden, Jim Lutine, we discovered that this plant is a member of the blueberry family, Comarostaphylus arbutoides. So Todd took it on to sequence DNA, plant DNA from the plant, fungal DNA from the mushroom, and then specific primers to isolate plant DNA and fungal DNA from the mycorrhizal roots. And then he compared those 4 isolations, and he found out that they matched exactly. So thanks to Todd, we were able to confirm that that lexinum was a mycorrhizal associate with this plant in the blueberry family. Another thing that Greg and I were curious about was we could see all of this diversity with our naked eyes, but Greg decided that we should try and do a specific sampling to see what kind of diversity occurred in a given area. So set up some plots at 2,400 meters elevation. It's primarily oak forest, and we contacted a plant taxonomist to help us determine how many different kinds of plants were there. And he basically said there's about 25. And we sampled these plots for 3 years, twice a year, once at the beginning of the mushroom season, and again at the end of the mushroom season. And over this 3-year period, we found that there were over 220 species of macrofungi. I mean, that's almost 10 times what the vascular plant thinks. but these are just macrofungi, the ones we could see. So we had no idea what kind of microfungi, molds and other things that we were in the habitat, but we couldn't determine what they were. Now, not all of these have been named yet, but clearly the diversity of macrofungi in this kind of environment is extremely diverse. And it was with the collaboration and serious efforts from Dr. Julieta Carranza at the Universidad de Costa Rica, who helped Greg and I push this project along for 10 years. And through the university and Julieta, we were able to train 3 students to study fungi through the university. And Juan Luis Mata went on to get his PhD in mycology at the University of Tennessee and is now a professor in Alabama. And as a result of our work on surveying the mushrooms in the Talamancas of Costa Rica, Greg and I wrote this field guide to about, there's a little over 100 species, the common things that we found described in this book on mushrooms in Costa Rica. Before Greg and I started in 1993, I had a chance, I was invited to go to tropical Queensland to look for mycorrhizal fungi. There were a number of people interested in mycorrhizal fungi of eucalyptus in Australia. And so there was funding to explore parts of Australia for mycorrhizal fungi associated with eucalypts. So in February '92, I had a chance to hook up with, from left to right, that's Mike Castellano, Jim Trappe, Neil Bowser, Ben Bayless, Paul Riddell, and the fellow in the blue t-shirt, Roy Young. And several years after this one trip on in northern tropical Queensland, there were 3 genera described, named for Mike, Jim, and Neil, and 2 additional genera described for Paul Riddell and Roy Young. Turns out all 5 of those genera are truffle-like fungi, not true truffles, but they grow in the same manner underground as true truffles. And I said to myself, after this happened, I have to get back to Australia sometime and study these things. So once Greg and I had finished up in Costa Rica, we went on about our business. Greg went on to do some other work, and I decided to come back to Australia. So I had the opportunity over the last 15 years to hit different places in Australia, New Guinea, Thailand, and Malaysia and New Zealand. Not all at the same time, but over a period of years, sampling different places. But the bulk of where I worked was in Northeast Queensland. One of the things that we found was a member of a true porcini. That is, it's related to Boletus edulis. But at the time, as far as I could tell, no true edulis-like mushroom had been found to grow natively in Australia. And we found this up in northern Queensland. And at the time, Bryn Dentinger was working on the true porcinis as part of his research project. And what he discovered using DNA DNA sequences and phylogenetic protocols, that it was basal to all of the other true porcinis known from the northern hemisphere. So this sort of flipped my whole view around about where is true diversity in porcinis. Well, it's not just in the northern hemisphere, but it's also in the southern hemisphere. And you can see in the top of this phylogram, there's another one from Thailand. Tucked in there, there's this other thing that has been placed in Xanthoconium and placed in Boletus that occurs in the United States. It's a not uncommon bolete-like mushroom, edulous-like mushroom that occurs in the eastern United States. And thanks to Walt Sturgeon, who let me use this slide, here's what Bolete separans, or Xanthoconium separans, looks like. Well, in northern Queensland, we found something that was like separans chemically, anatomically, and it reacted the same way. When you put a fresh drop of ammonia on the pigmented part of the mushroom, it turned this aquamarine turquoise blue. The downside of everything is that the spores and the hymenophore colors are all, recall, the morphological genus Tylopolis. And Todd Osmondson was with me on a trip to northern Queensland, and he did some rough gene sequencing from these things, and it turns out that it is also related to the true eduloids, the true porcinis. But we haven't published anything about this yet, and we haven't described this species. As I was going along on my own to work on these boletes from Australia, and gene phylogenies were becoming more and more mandatory for trying to understand where species belonged, I collaborated with David Hibbett and Manfred Binder to work on gene inferences and phylogenetics to try to understand, develop hypotheses for what was occurring in Australia. And Manfred is one of the first, or is the first person I know who actually did any gene sequencing for boletes when he did his PhD in Germany. And so these guys were really quite helpful in trying to sort out what was going on with boletes in Australia. One of the common big boletes that one encounters in Australia is this genus called Boletellus. And it's circumscribed in various ways, but no need to get into it at the moment. But the first one described from Australia was the Ananiceps, the first one listed here. And when you go and you look at the original description, trying to figure out what it is, you have 9.5 words in Latin that basically only describe the surface of the cap. That's it. So what the heck is this thing? And it wasn't 80 years later that Rolf Singer actually looked at the type specimen that came from Wangaratta in New South Wales. And Rolf said that it actually belongs in this genus Boletellus because it has longitudinally winged spores, but the wings don't have cross striae on them. So, all right, so now we have a little bit more information about what this species called Ananiceps is, described first in 1873. With Nigel Fechner, my collaborator at the Queensland Herbarium in Brisbane, we had the opportunity, with support from National Geographic, to explore mushrooms on Fraser Island, a World Heritage Site, the world's largest sand island. And while we were out there, we found this bolete that had thick, large flocosquamose warts on the cap that matched exactly what Berkeley described for Boletellus ananiceps. But you can see there's some additional features here that allow us to expand the concept of what this bolete was, and that there was pink pigment underneath those large warts. And if you look at and find young material, you can find that the pink color remains separate from this buff or brownish colored hyphae that's superficial to the pigment. And indeed, when you look at the spores under the microscope, you can see that there are wings or ridges, but there are no striae across those bands, those ribs. The other thing that we found was, oh, the stipe is white and the stipe stains brown when you bruise it. Oh, the tubes are yellow and they stain blue when you bruise them. And there's a veil that hangs down from the margin of the cap. So we're adding two expand the concept and refine the concept of this bolete. And when you cut it open, there's an intense, immediate blue reaction inside the flesh of the cap. But the blue reaction doesn't go all the way down into the stipe. It goes down partway. But the blue reaction happens so fast that you can't see the true color of the flesh of the mushroom. And it's actually yellow. And so I always try to encourage people that if you find one of these things, do a quick cut and look, because the blue happens so fast you don't see that there's a yellow unstaining before it stains. And down towards the base of the stipe, it gets this reddish brown. So none of this other information had been part of the original concept of ananiceps. Now we have an excellent concept that allows you to be able to distinguish it from the others that we saw in Australia. One of the ones that was very deceiving to us, because it incorporated characters of all the other scaly Boletella species that we were finding, and that's why we called it Deceptivus. And the DNA phylogenies supported its recognition as a distinct species. And when you scan the spores with the electron microscope, we found that these wings or ridges have these holes that go underneath the surface of the ribs. And I'm trying to figure out what the heck's going on here. Well, it turned out, in surfing the literature, Jacqueline Perrault in Paris back in the 60s had done some transmission electron microscopy, and she showed that in another species, different species that had the same kind of spore ornamentation, there were these translucent holes inside the ribs or wings of these spores. So when you look at the spore with the light microscope, what you're seeing is how light is bent and refracted when you look at through a light microscope. So what you're seeing are these holes that transverse the wings or ribs of the spores. And they're not actually ornamentation on top of the ribs of the spores. Absolutely incredible. Back in 1874, that shoemaker, Charles Frost, described this other icon from the northeastern US that he called Boletus chromopes. And distinctive, nothing else looks like it. Pink cap, bright yellow stipe base, and it has pink dots on the stipe, or sometimes there's a reticulum on the stipe. And over time in the 20th century, because of other characters, this species, chromopes, have been placed in 2 genera, and just based on morphological features. One was Lexinum, according to Rolf Singer, or was placed in Tylopolis, according to Smith and Tiers. Now, going halfway around the world into the southern hemisphere, we find a similar morphology. We've seen it in New South Wales and Queensland. It probably also occurs in Victoria, Tasmania. But what we thought, do we have an Australian chromopes here? Wasn't sure. A couple of guys, Neil Bowser and C.B. Wolf, found material, and they took on the concept that these were Tylopolis species, and so they described Tylopolis queenslandianus from northern Queensland and Tylopolis palomanus. Now you can see Queenslandianus had the dots on the stipe, palomanus has a reticulum. Now with working with Manfred Bender and David Hibbett, we did some sequencing, and we discovered that the Queensland stuff in the true chromopes, are neither even close to Tylopolis or Lexinum. And so we decided to describe 2 new genera to accommodate those 2 chrome yellow-footed boletes. But tucked in the middle here is this thing called Royungia. Now you remember back in the beginning, there was that fellow in the blue t-shirt, Roy Young. This mushroom, this fungus was named for Roy Young. And it turns out that it's an actual truffle-like bolete that grows underground. And so Royungia is genetically related to these chromapes type boletes that occur in Australia. And as far as we know, Royungia only occurs in Australia. The real find was working on Fraser Island was to discover this orange-red truffle-like fungus. I had no idea what it was. And because Jim Trappy is one of the world's gurus on truffle-like fungi, we sent it to him. And it turns out that he had collected it in northern Queensland, the Northern Territory, and Papua New Guinea. And we found it in northeastern Queensland and in southern Queensland. And it turned out to be a new genus of boletes that he called the sunset truffle. That's the name where the etymology of the genus Soleocassis comes from. It is beautiful. It's about the size of a ping pong ball or a golf ball. Beautiful fungus. And one of the most striking boletes that I had ever seen in Australia was this one that had a universal veil or a vulva. No other bolete in the family Boletaceae had this kind of feature, and this absolutely struck me. And working with one of my former students, Naveed Davudian, we were able to ascertain that it wasn't even in a porcini group or anything else, and it actually belonged in a separate subfamily and was distinctive, and it's separate genus and a new species. And we only just described this one last December in the series Fungal Planet. Now, I have to say that one of the best things about working at the Botanical Garden was the chance to work with students. And I had the chance to work with 4 PhD candidates, and Esperanza Franco went back to Colombia to be a professor at the Universidad de Antioquia. Maria Lise came from Brazil and is professor at the University in Florianopolis. Todd Osmundson is a professor at University of Wisconsin in La Crosse. And Naveed Davudian is a postdoc research associate at the Royal Botanic Garden, Victoria in Melbourne. And these 4 students really straightened me out and illuminated me on how bolete taxonomy really works. Lastly, I had the chance to work with a student from the Bronx High School of Science. She, part of their protocol there at that high school is in 3rd and 4th year students are asked to go pursue a science project outside of the high school. Well, Olga knew something about mycology since she'd been foraging for wild mushrooms with her family since she could walk. And she came to me and said, "Hey, do you have a mushroom project I could work on?" Well, I did. In fact, it was perfect timing because we had found this bolete. We knew about this bolete that occurred in Southeast Asia, first described from Singapore in 1909, but it didn't seem to fit anything that we knew about. So Olga did work in, basic historical bibliography to try to figure out what was going on, what this thing was, who described it, where it grew. She learned mycological techniques for examining mushroom specimens. She learned DNA techniques for isolating, sequencing, and reading DNA sequences and getting a phylogeny going. And eventually, it all came down to publishing this paper that in 2019 for a new genus that included the Boletus longipes from Singapore and another species of the same genus that we were finding in Australia. Now, what Olga did was she submitted her research to Regeneron competition in the USA. She came out as a semi-finalist scholar in that competition, and she submitted it to the New York State Science and Engineering Microbiology competition, and she got first prize. Yay! Bolete taxonomy can take prizes in science fairs. Absolutely fantastic. Lastly, I want to say thanks to my wife and daughter for their support, guidance, patience, humility, for letting me do this kind of work for the last 35 years. Thanks so much. I appreciate it.
Wow, Roy, thank you so much. That was really a fantastic presentation, and we've had so much great conversation happening in the chat. You have some collaborators here in the audience who are very happy to see the research and share some of their own wisdom. And people are literally falling in love with your photos of mushrooms. So it's really great. And just as a reminder for everyone in the audience, please go ahead and submit your questions to the Q&A. And you can actually upvote the questions others have already submitted, and that will help us to narrow things down because we only have a limited amount of time today. So I'm going to go ahead and get things started off.
Sure, John.
So a question came up from our audience that was has a lot of interest. When the species were considered unknown, is that always true just for Western scientists? And was there contact with indigenous peoples in these regions that you studied to find their knowledge and familiarity with these species?
Part of the permitting process in Australia requires that you contact indigenous tribes or linguistic groups for permission to collect in their areas. And as near as I could tell, as far as boletes go, there weren't any input into what grew there, and none of these were known. So as far as I know, the indigenous folks didn't have any input onto boletes. I know that there are some things that the indigenous folks knew about and would eat, but as far as I can tell, there weren't any boletes in there.
Thank you. What was, this is coming from one of our audience members, what or where was the strangest habitat or the most unexpected habitat where you found a bolete?
In the sand. I mean, here was this World Heritage Site called Fraser Island, which is about 3 hours drive north of Brisbane. It's the world's largest sand island. And at the time, they knew all the plants. There was a census of all the plants and all the animals that occurred in this World Heritage Sites, but there were only 2 records of fungi from this place. And so I got a grant from National Geographic to survey that, and there were mushrooms everywhere. And I said, what are they doing growing in the sand? Well, it turns out that in order for that forest to survive in sand, they have to be associated with fungi. And so over a 3-year period, Nigel Fechner and I were able to document, well, we made 7 trips to Fraser, and I forget how many specimens we've collected, but some of those things are just totally unknown before. But that was truly one of the weirdest places, just to see mushrooms growing in sand. And that, good God, that's nuts.
Wow. And those were mycorrhizal associations, right?
Exactly. Mycorrhizal associates. Yeah. But not always. I mean, there's wood rotters there as well, but that's the only way that these trees in the eucalypt family can survive, is with the mycorrhizal root association.
Thank you. I know you told one story already about eating a new species that you discovered. Our audience thought that was very funny. So how do mushroom experts like you, though, go about deciding whether or not something is safe to try or edible rather than being poisonous?
Well, if I always try to tell people, if you know the genus, like a chanterelle, and chanterelles occur in Australia, but to my knowledge, nobody has eaten them, at least the people that I've talked to. And so I say, if you're going to try, this is a chanterelle, and if you're going to try it, just eat one and wait 24 hours and save some in the refrigerator in case you get sick, so that you can show the poison control people, the people at the hospital, that this is what you ate. But everybody reacts differently to edible, or what are called edible fungi. For example, that Lexina monticola from Costa Rica that Greg and I described, that grew with the Comaro staphylos. We said one day there was just heaps of it, and I said, "Hey, Greg, I mean, Lexinum's edible. Let's see if the cook down at the cabins where we stay, if she'll cook them up." And he goes, "Yeah, okay." So we made deliberately collections to go on the table, and I ate them. I was fine. Greg's wife ate them. She was fine. But the next morning, he was stooped over and he was accusing me of poisoning him. And so Greg didn't react well to eating those things. So, and again, it's an idiosyncratic thing. I try to tell people, if you're going to do it, take precautions.
Yeah. So it sounds like there's some inherent risk involved, no matter how much expertise you have.
Exactly. Exactly.
Yeah. We have a question about mycoremediation, which is not something we covered today, but if you're familiar with that process, is basically saying that if mushrooms are being used for mycoremediation, like removing heavy metals, uranium, chromium, do mushrooms leave those products behind in the soil? Do they truly leach them out and allow them to be removed? What's your understanding of that process?
I mean, I've heard of it, basically the work that Paul Stamets has done on this kind of thing, but I haven't pursued it, in detail to see what happens with these toxins once they're pulled out of the environment. Where do they go? It's something that I've wondered about myself, but I've just haven't gone along to try to figure out what's going on there.
Great, thank you. Thank you, Brian.
Sure. Yeah.
Have any medical applications been discovered in this group of fungi?
Medical. Just off the top of my head, I can't think of anything. But that doesn't mean that they're not there.
And on that topic,
yeah,
any hallucinogenic properties? People are really interested in these things for boletes. Is there properties that boletes in general are known for?
I don't know about any hallucinogenic boletes. Oh, wait. Yes, I do. There was a report from New Guinea. There was a French mycologist. His name was Roger M. And he was collecting up in the mountains in New Guinea, and he found this bolete that the local folks would eat to get high. And so the species was undescribed, so he called it Boletus manicus. And yeah, so and I've never seen it. When I was in New Guinea, I didn't see it. And I don't know of any further reports other than that original one by Roger M. about this supposedly hallucinogenic bolete. That's the only one I know of, John.
Interesting, but it sounds like there are a fair number of edible ones in the family at large.
Yeah, and in the last 10, 15 years, more people are eating boletes. And I remember one case where a mycologist, and people will know him, Gary Linkoff, would eat this bolete. And he said he ate it about 8 or 9 different times. But as time went on, he didn't feel as good, so there was some kind of an accumulation of something from the bolete over time, so that he developed that. He says, I can't eat this anymore because it makes me sick. So I I'm not sure what's going on there.
Interesting.
Yeah.
So we had a question come in that actually a lot of our participants have weighed in on, but I'd be interested to hear your perspective on the answer as well.
Sure.
How can hobbyists and other private citizens contribute to what is going on in academia? And vice versa, how can people in labs make the best use of willing volunteers in the public?
Well, I know that here in the U.S., there's a group that started out calling themselves. They were involved in a project called mycoflora. Now it's called fungal distribution, or Fundis. And so what they're trying to do is to get people to contribute well-documented specimens that can be used for DNA analysis, and and voucher specimens deposited in herbaria in the U.S., and develop a general idea of what occurs in the U.S. And I know in Australia, for example, there's a group called Fungi Map, and they've been doing this for at least, well, when I first went there in 2005, they've been going on for a couple of years. And they've started to move all of their data to this app called iNaturalist. And so that way they can map what they know about macrofungi in Australia. And so sometimes it's just visual observation, other times it's actual physical specimens being deposited at local herbaria. And there are groups up and down the coast, I know, in New South Wales and in Queensland, where people are involved in collecting and vouchering specimens. And I've been in touch with people in New South Wales, Tasmania, and Queensland about some of these things. And occasionally I get a voucher specimen to be able to work with. Yeah. Actually, in 2020, we published another paper of a bolete that only occurred in Western Australia. And so we were able to, with the collaboration of this woman in Western Australia, be able to document that this thing existed out there. And without her collaboration, we would have never gotten it on in press.
Wow,
So
that's a great story. Fundus, someone else mentioned that as well, and also iNaturalist was mentioned in the chat as ways that citizen scientists can participate, collecting data that can be used and published on. And also mentioning that, if you're collecting plants, make sure that you talk to your local herbarium, make sure that you're doing things correctly, plants and fungi, I should say. And because there's really important rules to where you can collect and where you can't, and who you need to attribute and all that stuff. So just make sure you're in touch with someone in your area who's willing to help with that process.
Yeah, exactly.
Michael asks, bolete genera have changed so much in recent years. What is the best way to get up to speed with the new names and genus concepts?
Gee, that's tough. It's hard for me to keep up, because when I started out, there was maybe 12 to 20 genera, and now there's well over 87 that have been documented using DNA. So it's exponential what's going on. It's really hard. There's no easy way to keep up with what's going on. I maintain this website. I don't know if you're going to tell the people who are listening about it, but I try to keep up with the new names and some generic synopses on that bolete website about what's going on, and I try to update it at least twice a year to bring it up to date. And there's also some guides there on how to document, describe, and properly prepare material for scientific study, if that's what they're interested in. But there's no looking at that online site called Index Fungorum is useful up to a point, but it's not necessarily completely accurate. Well, we always find errors there once in a while, and the man who runs the site is quite willing to make changes as we see them.
Wonderful. Thank you, Roy. I just shared that link in the chat to the NYBG Bolete webpage if anyone is interested in that. And as you mentioned, there's also literature about preservation of fungi collections, which was another question that came up.
Yeah, there's a couple of PDFs there that they should be able to get for free and have a look at.
Great. We're running out of time. I'm just have time for one more question. And this comes from Noor. I'm new to macrofungi. Before this, I was into microfungi. But when I discovered the beauty of macrofungi, I found in my university forest, I got hooked. Could you suggest any books for a newbie like me, books that can help with identification, particular boletes, but macrofungi in general? That would be really appreciated.
Yeah, there's a number of field guides out there right now that have come out in the last 10 years or so. One on boletes. There are two authors, Arlene and Alan Bassett, have written a couple of books on North American boletes. Tim Baroni has written a book on Northeastern macrofungi, and I think it's a Timber Press book, but I can't remember for sure. But that only came out a couple of years ago. What was the guy's name? Todd Elliott and Steve Stevenson wrote one for Southeastern US mushrooms, and they're quite good. They're really, really good, quite informative, and they show you what they found and what they recognize, but there's also some other general introductory things there as well.
Terrific. Thank you, Roy.
Sure.
We had some other chat comments come in with suggestions from our audience, which is really helpful. So if you get a chance to check out that chat at the end before you leave, there's lots of great resources people have been posting there. So thank you all for doing that. All right. So on that note, we always have more questions than we have time to answer. So Roy, thank you so much for sticking around and helping to work us through it all. Thank you so much for that talk. We learned so much. It was really beautiful. So much about fungi. So thank you so much for doing all that.
Thanks for listening. Thanks for hosting.
Great. And thank you to our audience, everyone who's here to join today's presentation. If you missed any part of this event or wish to watch it again, a recording will be saved and shared on the NYBG lecture library. So we hope you'll join us for future science, conservation, and humanities lectures from the New York Botanical Garden. Next Friday, our colloquium, Humanistic Uses of Herbaria, held in partnership with Dumbarton Oaks, invites 4 experts to discuss innovative uses of herbarium specimens at the intersection of history, science, and art. And on April 2nd, we'll be celebrating daffodils, tracing the history of rare Narcissus cultivars from Europe to the United States with horticultural specialist Sarah Van Beck and New York Botanical Garden curator Claire Lyman. So for those who haven't already registered for our mailing list, go ahead and sign up using the link in the chat that I'll post just in a moment. So until next time, please stay well, stay green, and take care. Goodbye, everyone.